Did everybody get the email I sent out yesterday about the revision and the breakpoint? Okay. I made a little adjustment because otherwise, if I stopped where I said I was going to yesterday, that's going to make the second exam pretty long. So there's kind of a natural breakpoint, and you will get there probably early Thursday. So not that much difference. Jill Novotny had asked me to request that you, at least if you're not ready to start trying to do making medicine yet, to at least log on to make sure that you can log on. Because evidently there have been a couple of people who have had some trouble logging on. And she said to resend you the link. If you haven't gotten the email because it's in your spam, email Jill Novotny and ask her to resend the link to you. I know some people have gotten on it or are working on it, but at least try logging on to make sure that you can do that. Okay, tomorrow in lab, we're going to be doing 3 staining techniques. We're going to be doing a simple positive stain, a simple negative stain, and most importantly, a gram stain. And the gram stain is going to be really crucial in terms of identification of your unknown. Gram stain is a stain you will do over and over again. And I already told you that that's one of the things you will have to do for memory on the lab exam. When we're working with stains, please wear something to lab tomorrow that is either expendable, or when you come in, go in the back room back there and grab a lab coat, because once you get stain on clothing, it is very, very difficult, if not impossible, to get out. If you get it on your hands, it will eventually wash off. If you don't want to walk around with purple hands or something, you can come over here and grab some gloves during the lab tomorrow. Colored hands are kind of a badge of honor for the microbiology lab. Make sure you read through the lab tomorrow ahead of time. If you know what you're doing, it is much easier to be strategic and kind of plan out the order and timing in which you do things, and that will make sure that you get done in time. I think that's everything you need to know as far as tomorrow is concerned. Questions? Okay, let's finish going through Berge's manual and talking about the last 2 volumes. We won't spend much time on that, and then we will immediately transition over into the next topic, which has to do with growth of microorganisms. So we're at volume number 4 right now, okay? How does volume number 4 deal with, or differ from volume number 3? The GC content of these bacteria, okay? So these are the high GC content bacteria, greater than 50%, and they are gram-positive bacteria as well. In terms of taxa, pretty easy to deal with. A single phylum, Actinobacteria, and a single class within that phyla, also Actinobacteria. So not much to remember there in terms of taxonomic groups. There are some genera within that class Actinobacteria that I do want to briefly mention to you. One of the genera within that class is the genera Actinomyces. These are interesting bacteria. These particular bacteria are oftentimes referred to as the branched bacteria. And although they're smaller, morphologically, they look a lot like fungi. Did you look at fungi in botany? I think you did, didn't you? And one of the things you saw with fungi is that part of the life cycle of the fungi typically includes a filamentous stage, where these long branched filaments, called hyphae, make up the majority of the structure of the fungi. The fruiting body of the fungi is oftentimes just a mass of these intertwined hyphae. So that's what a mushroom is. It's a whole bunch of hyphae packed together to form this fruiting body. Well, these Actinomyces kind of look like these fungal hyphae, but remember, fungi are eukaryotic, and these are bacteria, so they're prokaryotic. These particular bacteria are found very commonly in soil. And if you ever pick up nice and rich soil and smell it, oftentimes soil smells kind of musty. And that musty smell of soil is typically due to the metabolic activity of all of these actinomycetes that are found within that particular environment. Another genus within that particular class is the genus Streptomyces. And the only reason I'm going to mention this particular genus right now is because these are bacillus bacteria that can produce a variety of different types of antibiotics. You may have heard of the antibiotic streptomycin before. Streptomycin is produced by this genus of bacteria, Streptomyces. One of the things we're going to talk about when we talk about antibiotics later on as a medical treatment against bacterial infections is the fact that resistance is evolving against many antibiotics. And pharmaceutical companies that want to develop new antibiotics are constantly looking for new sources of antibiotics. And the sources of the antibiotics are bacteria and fungi. Streptomyces are also very common in soil environments. And so one of the things that pharmaceutical companies have started to decide is protecting habitat where these soil bacteria live might be an important strategy in trying to ensure that there's a continued supply of these life-essential drugs to treat diseases. So by protecting habitats, particularly in unique environments by tropical rainforests, they're hoping that they will protect the environments where these bacteria live and later be able to go back and kind of mine those habitats for the organisms that can produce these particular drugs. So there's a place where conservation and business really do go hand in hand. Medically speaking, there's a very important genus within Volume 4, and that is the genus Mycobacterium. And I've mentioned these already in a couple of other contexts, specifically the species Tetani and Lepri. So Mycobacterium lepri causes leprosy, and Mycobacterium tetani causes tetanus. We'll talk about the tetanus toxin later on in a fair amount of detail, but I want to spend just a couple of minutes kind of familiarizing you with Mycobacterium lepri and, whoops, sorry, made a mistake. Not thinking this morning. It's tuberculosis, not Mycobacterium tetani. Clostridium is a tetani. So two medically important bacteria within this genus Mycobacterium, Mycobacterium tuberculosis and Mycobacterium lepri. We've mentioned Mycobacterium already. We mentioned Mycobacterium in the context of a structural feature that certain species of bacteria have, including this genus, and that is these organisms have in their cell wall a fatty substance called mycolic acid. Mycolic acid can be detected in cell walls by doing a stain called the acid-fast stain. And we will do an acid-fast stain. So both Mycobacterium tuberculosis and Mycobacterium lepri are acid-fast positive bacteria. And that's not that common. And so this becomes a fairly easy, straightforward way to diagnose these two particular diseases, particularly in their early stages. Tuberculosis is something that is making a comeback right now, unfortunately. For several decades prior to this, tuberculosis you just didn't hear much about, because once antibiotics were developed, there was an effective method of treating this particular disease. However, in the pre-antibiotic era, tuberculosis was a huge public health problem in this country and worldwide. Particularly in the mid to late 1800s, tuberculosis was rampant in this country. And one of the characteristics of tuberculosis is if you inhale the bacteria that cause this particular disease, they can start growing in your lungs. And one of your body's responses to this particular infection is forming this tough capsule around the infectious organism called a tubercle. And those tubercles can contain live tuberculosis bacteria, which can then infect other people if you cough those up and breathe on them. Tubercles are easily identified in chest x-rays, and so it's pretty straightforward. If you've got this hacking cough that you can't get rid of, and your doctor takes a sputum sample and says, oh, acid-fast bacteria. If you do a chest x-ray and see those tubercles, then that's pretty definitive evidence of the fact that you have tuberculosis. Like I said, prior to antibiotics, tuberculosis was a real scourge in this country. And about the only thing that they really knew how to do, and this was just mildly effective, was to isolate people and particularly take them to areas like the far west or the mountains where they felt the air was cleaner and have people live there for long periods of time. There were tuberculosis sanatoriums, which were facilities that were dedicated to taking care of people who had tuberculosis. And once you lived in a sanatorium, the likelihood that you would ever get out of it was extremely poor. Unfortunately, there are multi-drug resistant forms of tuberculosis that are now emerging, and they seem to be emerging most commonly in areas of high poverty, where people aren't living in sanitary conditions, and people are all packed together, particularly in slums in underdeveloped countries, where the likelihood of transmission of this disease is pretty high. And so tuberculosis is one of those re-emerging diseases that we thought we'd kind of dealt with, but is now becoming an increasing public health threat. Mycobacterium leprae causes, anybody had a tuberculin skin test? Most people will probably have to have one if you find yourself wanting to work in any kind of healthcare environment. Did they require you to do TB tests for the JMH winter term? Okay. Yeah, I couldn't remember if they required you to do that or not. The tuberculin test is actually a little test where they scratch your skin with some of the antigen of the tuberculosis bacteria. And what we're really looking for is to see if your body develops an immune response. It's called a delayed hypersensitive reaction against those particular antigens. The only way you can develop a delayed hypersensitive reaction is to have antibodies against tuberculosis. And the only way that you can have antibodies is if you would have had a prior exposure or infection. And so this particular tuberculin skin test is based upon, have you been priorly exposed to that particular bacteria? Mycobacterium leprae, causative agent of leprosy. Leprosy is not something you hear much about anymore. Leprosy was probably most commonly encountered by people if they read the Bible and read about biblical times, because leprosy was a real threat then. One of the things that happens in leprosy is that they decided that they needed to quarantine people. So they developed leper colonies, for example, where people were put because there was such a fear that leprosy could be transmitted from one individual to another. And leprosy is contagious. But as it turns out, leprosy is one of the least contagious diseases. And so the idea of putting people in leper colonies was probably a big overreaction. But it was such a fearful disease, and it had such fearful consequences. Loss of fingers, toes, face lesions. One of the reasons that the extremities of the body are affected most by leprosy is that these particular bacteria have a temperature optimum that is pretty narrow. And they prefer a little bit cooler temperature than most other bacteria. And the extremities, like the fingers and the face and the toes, tend to naturally be a little bit cooler than your core body temperature. And so those are the parts of the body where those leprosy bacteria tend to live and then cause the damage to that particular tissue. Leprosy also has something that we'll refer to in probably later today, a very long generation time. And that means that it is very slowly reproducing. That's one of the reasons why it is not particularly contagious. The last leper colony was in Hawaii, and I think that leper colony persisted until the mid to late 1800s. And if you go over to Molokai, which is one of the islands on Hawaii, you can still tour the remnants of the last existing leper colony. So leprosy, not as big of a public health threat as it once was, obviously. Tuberculosis, yes, it is reemerging. There's one more genus within this particular volume that I will just mention, and that is the genus Micrococcus. Micrococcus looks a lot like Staphylococcus. These are clusters of cocci, and they tend to be found in groups of 4 called tetrads. Micrococcus, Streptococcus, and Staphylococcus all used to be grouped together in earlier editions of Bergey's Manual, because why? They have a similar morphology. Okay, they're all cocci, and therefore people assumed that they were evolutionarily related, that they had a lot of common ancestry. Well, now we saw last time that Streptococcus and Staphylococcus are now within volume 3 within this class Bacillus, and then in a totally different volume, here's some more cocci that are in an entirely different volume, an entirely different evolutionary group. And so this again does illustrate how poor morphological shape Bacillus cocci spirillum is in terms of being a good indicator of phylogeny or evolutionary relationship. The last volume of Bergey's Manual is one of these kind of catch-all groups and contains several different types of organisms. You'll notice that it is divided into only phyla, for example. And so that's one of the ways that it differs from the other 4 volumes. The other 4 volumes contain at least 1 or more classes. This one doesn't. Okay. There are 2 phylum in here that I just want to call your attention to because we will be referring to those later on. And they are the phylum Chlamydia and the phylum Spirochetes. So last volume, volume 5, a couple of selected phyla. The first being the phylum Chlamydia. Within the phylum Chlamydia is also the genus Chlamydia. And within that genus are several species of chlamydia that are pretty nasty pathogens. Chlamydia are really small. So like rickettsia, their small size is one of their outstanding characteristics. These are also, like rickettsia, obligate intercellular parasites. So they have to be inside of living cells to function. What's their limitation? Well, they've got a big limitation. They can't make their own ATP. So really, chlamydia are kind of energy parasites of other cells. When we talked about rickettsia, we said that one of the things that was important about rickettsia is that they required a vector to be transferred from one organism to another. And the vectors were arthropods like ticks and fleas and mites. Chlamydia don't require a vector. Typically, the primary way in which chlamydia are transmitted is sexually. They're a venereal disease, and so they are a sexually transmitted disease. And as it turns out, this is one of the most common sexually transmitted diseases, and is oftentimes completely missed, and it is very much underdiagnosed. What's the consequence if you have chlamydia, particularly if you're a female? Well, one of the consequences can be reproductive failure. And chlamydia can cause scarring of the fallopian tubes, and as a result, conception becomes very difficult. Chlamydia can also be transmitted to a newborn as they move through the birth canal, and chlamydial eye infections can occur. And this is what a nasty chlamydial eye infection of a newborn looks like, whose mother was undiagnosed and had a chlamydial infection, and then the baby picked that up as they were being born. As the realization occurred that these chlamydial infections were becoming a problem, one of the things that was commonly done with newborns was to prophylactically or preventatively treat them with something that would kill the chlamydia. And the first thing that was used was to put silver nitrate drops in the infant's eyes. Anybody ever gotten silver nitrate on your hands in chemistry? Causes your skin to kind of turn brown, and it burns. Okay? So you can imagine the discomfort that newborn infants suffered when they put these silver nitrate drops in their eyes. Now, probably better than this outcome. Okay? Silver nitrate treatment has now pretty much been replaced by antibiotic drops like erythromycin, and obviously those aren't nearly as irritating as the silver nitrate. A second phylum that's of interest in this volume is the phylum Spirochetes. And these particular bacteria are the spiral-shaped bacteria. They have that spirillum shaped. And you may not remember this term, but spirochetes are also characterized by having an axial flagellum. A little brief reminder. In axial flagella, the flagella wrap around the body of the cell and kind of are inserted in kind of a groove in the surface of the cell. And as these axial flagella move, it causes a twisting, kind of corkscrewing type of motion. And these bacteria can actually burrow then into tissues. And that's oftentimes part of their pathology. One of the genera within this phylum Spirochetes is the genera Treponema. And the species Palladium is one that is of a lot of medical interest. Not quite as much as it used to be, but still a fair amount of medical interest. Treponema palladium is the causative agent of the sexually transmitted disease syphilis. So this is the syphilis organism. So here's Treponema palladium. And you can see these spiral-shaped cells, these spirochetes. Syphilis is a very insidious disease. Now, again, in the pre-antibiotic era, syphilis was a huge public health problem. Post-antibiotic era, not as much, although again, making a comeback, particularly because of drug-resistant forms of syphilis now having evolved. Syphilis has kind of an interesting history. It can go through several different stages of infection. They're called the primary stage, the secondary stage, and the tertiary stage. And there can be long periods of latency in between when you show symptoms for those particular stages. The first stage, or the primary stage, usually manifests itself as blistering. That's called a chancre. Most common locations for that are on the genitalia, also can be on the lips, and oftentimes this particular manifestation resolves itself pretty quickly, and the person thinks, okay, I'm okay, it's over. But typically those bacteria then go into latency and can eventually, years later, break out in a secondary syphilitic rash. And the rash can be all over your entire body. Okay, that would get your attention, right? But even this secondary syphilis can abate. So again, you might think, well, okay, I made it, I'm okay. But then years later, tertiary syphilis can result, and tertiary syphilis can result in neurological damage and also erosion of tissue. The tissue kind of starts to fall apart. And tertiary syphilis is deadly. The notorious gangster Al Capone died from untreated tertiary syphilis. So this bacteria is not anything to mess around with. Syphilis was such a scourge that this was one of the first diseases that people tried to develop a selective treatment against. We're going to talk about that concept of selective treatment later on. But selective treatment involves, can I develop a drug, a quote-unquote magic bullet, which will kill the pathogen and not the host? Well, antibiotics are great selective treatments. They're great magic bullets. But what did they use prior to the development of antibiotics? There were all kinds of drugs that were tried. One of the drugs that was marketed for syphilis was a drug called salvarsan. And one of the earlier pioneers in chemotherapy, a guy by the name of Paul Ehrlich, developed this compound, salvarsan, to try to treat syphilis. The name salvarsan indicates that this treatment was supposed to give you relief or salvation from the scourge of syphilis. What was the drug? The drug was an arsenic-based compound. And what do we know about arsenic? It's a poison, right? Okay. So many people argued that this first chemotherapeutic drug that was developed, salvarsan, was probably worse than the disease in many cases. Yeah, it killed the bacteria, but it caused tremendous damage to the person taking it as well. Neurological damage in particular. Another genus within that phylum of Spirochetes is the genus Borrelia. And the species Burgdorfii is particularly interesting. Borrelia burgdorfii is the causative agent of Lyme disease. And Lyme disease is something that we're hearing a lot about. And increasingly we're hearing about it in parts of the country where it hasn't existed before. Here's the spirochete. Here's the Borrelia spirochete. So you can see that helical shape, characteristic of spirochetes. Borrelia is transmitted by a vector, and in this particular case, it's a tick. And the tick transmits the bacterium from its natural host, which is the white-tailed deer, to humans. And one of the characteristics of a Lyme disease infection is what you call this bullet rash. Lyme disease is pretty insidious. Some people have Lyme disease infections and the symptoms are pretty mild. It's kind of like a flu-like symptoms. But some people respond very adversely, and there can be tremendous inflammation causing severe joint pain. Lyme disease in some individuals can be really debilitating. And it doesn't always give you this nice, easily identifiable bullet rash that physicians easily can identify. And so Lyme disease is one of the diseases that is oftentimes totally missed and underreported and misdiagnosed as something like rheumatoid arthritis, for example. Okay? The problem with that misdiagnosis is Lyme disease is treatable by antibiotics. Obviously, rheumatoid arthritis is not. So you need to know what your disease is to be able to treat it properly. Lyme disease used to be primarily endemic to the northeast part of the country, Vermont, New Hampshire, Maine, etc. Because that was the primary location where you found the host, the white-tailed deer. But over a period of time, particularly with more urbanization, loss of habitat, climate change, the range of the white-tailed deer has been increasing, and now it's common to find them in states like Indiana, where they didn't exist before. And so now, Lyme disease has spread to surrounding states because of that migration of the host for that particular pathogen, the white-tailed deer. And so that's been an interesting epidemiological study to look at the spread of Lyme disease and the factors that have contributed to the spread of that particular disease. Okay, so that's our overview of Berge's Manual, the 5th edition. What my hope is that you have a little bit better sense right now of how, at least presently, these organisms are grouped and what some of the criteria are for putting organisms into different groupings. Different groupings are indicated by different volumes, obviously. And then what some interesting organisms are that you may have heard of before or that we may encounter again, and where they fall within this particular grouping. So one of the things people are always interested in is some kind of bottom line. What is some of the basic taxonomic stuff that you have to know? Well, there's not much you need to know about Berge's Systematics 1st edition. One of the things I do want you to know are the names of those 4 divisions in the kingdom Monera: Lichides, Tenerichides, Gracillichides. And what are the criteria for those divisions? They're primarily based upon cell wall structure: gram-positive, gram-negative, no cell wall, or RK bacteria. Okay. For the 2nd edition, what are the names of the 5 volumes and what domains do they include? Well, that's pretty easy because 4 of the 5 only have the domain bacteria. It's really just the 1st volume that has both the RKs and the bacterial domains. I don't expect you to know the names of all the phyla and classes in each of the volumes, but I would like you to know the numbers of them, okay? So you have kind of a relative understanding of which have a lot of phyla and classes and which have a few, okay? And then, for these representative organisms that we've talked about, where do they fall in this classification system? And so, can you assign them to a volume? Okay? Within that volume, can you assign them to a phylum? And if I talked about it, a class or a genus. So we're only going to concentrate on those selected examples that I gave And if I didn't talk about a phylum and give you an example of it, then you're not responsible for it. So right here, for example, in this first volume, we only talked about these two phyla. Okay? In this volume, we really only talked about 3 of these classes. Alpha, gamma. Really only talked about 2 of them. Alpha, beta, gamma, sorry. So it's not quite as daunting as it might seem. Okay, so that's enough taxonomy. So now let's get into some practical information. And what we're going to talk about is growth characteristics of bacteria. One of the things that I think that is extremely important to appreciate about bacteria is their tremendous growth potential. And one of the ways in which we can measure the growth potential of an organism is to make reference to its generation time. How long does it take the organism to produce offspring? Obviously, the shorter the generation time, the greater the growth potential of the organism. Now, when we talked about bacterial cells and the fact that they are prokaryotic, one of the things we concentrated on was the fact that prokaryotes are small, and small organisms typically have a fairly fast metabolic rate. And one of the reasons they can have a fast metabolic rate is that they have a very favorable surface area to volume ratio. So small organisms, favorable surface area to volume ratio, high potential for dividing quickly. Average generation time for a bacteria is about 20 minutes, although that figure is highly variable depending upon the species, but that's kind of a general measure. Bacteria reproduce by a very simple process, binary fission. And binary fission is illustrated right here. We have a bacterial cell with a single circular chromosome. At some point in time, that genetic information duplicates. So it has 2 single circular chromosomes. We can have an invagination or an in-pocketing of the cell membrane. A mesosome can form. And that mesosome can attach to these particular chromosomes and kind of anchor them so that as the cytoplasm pulls away, that helps to segregate the 2 chromosomes into different halves of the cell. The cytoplasm divides, and eventually the cell divides in 2. And in about 20 minutes, we have 2 cells that are identical to the original parent cell. The cell literally has split in half. Binary fission. Now, when we talk about growth of most organisms, typically what we're referring to is the growth of an individual, and particularly an increase in its mass. Okay? So when we talk about your growth, and if your parents chart your growth, they weigh you, they measure you, and that is the way growth is expressed. When we talk about bacteria, however, typically we're not talking about changes in mass of an individual cell. because it's really hard to measure changes in mass of individual bacterial cells. Yes, these cells will probably get a little bit bigger prior to dividing again. But when we talk about bacterial growth, typically that's not the attribute we're talking about. When we talk about bacteria, what we're typically talking about when we talk about growth is increase in the numbers within a population. So we measure cell numbers rather than cell masses. Let me give you a practical example of how we can go about calculating bacterial growth within populations and some of the implications of those calculations. So let's say that I've got an egg salad sandwich. And it's contaminated with some staph bacteria. It's not uncommon, okay? Eggs typically can pick up bacterial contamination, and staph is one of the bacteria that's not uncommonly found in egg-based materials. Well, what if I started out with just a very few staph cells? About 10 of them. If I leave that egg salad sandwich out at the temperature at which those bacteria can divide, how many am I going to have after 4 hours? To make this calculation, I'm going to have to know one other piece of information. I'm going to have to know the generation time of staph. And it's approximately 20 minutes. Okay, so how can I make that calculation? How can I make that determination of how many cells I'm going to have if I start out with 10 and I have a generation time of 20 minutes and I let them grow for 4 hours? How can I measure the change in that particular population? Well, there's a simple formula. N sub f equals N sub I times 2 to the nth. N sub f is the final number of cells. That's what I'm trying to solve for. So how many am I going to have after 4 hours? N sub I, how many do I start out with? 2 to the nth is the number of generations. And is the generation time. And we have determined that our generation time is 20 minutes. So if you do this calculation and you plug the numbers in, after 4 hours, there are about 41,000 Staphylococcus bacteria. If we let this population grow for an additional hour and the total time is 5 hours, total number of cells pops up to about 327,000. So we're getting a significant amount of growth in a short period of time, and that growth is rapidly accelerating. Look at the rate at which growth changes over a period of only a single hour. That's characteristic of a type of growth called exponential growth. Exponential growth is clearly illustrated here, where we have one cell dividing to form 2, 2 to form 4, 4 to 8, 8 to 16, 16 to 32. So the number of generations grows exponentially, and we see a rapidly increasing rate of growth over a relatively short period of time. If we plot cells versus time, if we plot the actual number of cells versus time, exponential growth manifests itself as this type of a growth pattern. This is called a J-curve. And one of the things that you can see is that initially, growth rate increases fairly slow, but then once you get around that bend, things really start to take off. Well, that kind of makes sense when you start looking at this exponential growth rate pattern, okay? It takes a while for the number of doublings to have a significance, but eventually it gets really, really fast, really, really quick. If we plot the log of the number of cells versus time, then we get a straight line. Well, what I want to do next time is take a look at the typical growth pattern that we see within microbial populations. So we're going to look at a plot of the log of number of cells versus time and see that growth curves typically don't have just this straight shape, but there are some nuances to how bacterial populations grow. So we're going to talk about the growth curve. Okay?